Hello everyone, and welcome to this audio podcast. In this series, the LabZtorm effect v.3. This is an audio podcast aligned to the role of endogenous opioids in ischaemic preconditioning or IPC in humans. This will be an audio podcast in line to Beta-Endorphin, Beta-Endorphin in context of cardio protection, and also in a line to remote ischaemic preconditioning and ischaemic preconditioning in humans. This is another audio podcast aligned to a Storm Lobostics soliloquy and another Storm Lobostics production. So in following the audio podcast aligned to endogenous opioid peptides or EOPs in remote ischaemic preconditioning, and here in this audio podcast we're going to delve into beta endorphins more specifically. So the hypothalamic pituitary axis or HPA axis is believed to play a primary or a pivotal role in the body in context of physiology and the body's reaction to stress by balancing hormone secretion from the adrenal medulla and the adrenal cortex. Beta endorphin beta endorphin is documented as a neuro has been documented and evidence as a neurohormone comprising 31 amino acids. It is a sort of known has been documented as a cleavage product of opioid melanocortin or POMC cells in the arcuate nucleus within the hypothalamus, and in a small population of neurons in the lower brainstem. So, for example, the medulla oblongata. POMC cells, so pro-opiomelanocortin POMC cells are also the basis or precursor for adrenocorticotropic hormone or ACTH. And so beta endorphin is also aligned to this, as beta endorphin is also an endogenous opioid peptide, which is secreted by the pituitary gland and reaches all tissues present in the body by diffusion. Beta endorphin, as well as the messenger RNA or mRNA for its precursor POMC, are not only found in pituitary gland but also within various types of immune cells infiltrating inflamed subcutaneous tissue. Beta endorphin is localized secretory granules packed in membranous structures in macrophages, monocytes, granulocytes, and lymphocytes. During stressful stimuli, beta endorphin is released and interacts or interplays with peripheral receptors, specific peripheral receptors to inhibit pain by blocking a signal to the nervous system. Then works by Gross Gross in around 2003. I had highlighted and suggested that enkephalins are perhaps the most likely endogenous opioid peptides, which serve as triggers and distal effectors of IPC in laboratory rabbit models. It works again by Pearl and team in 2005, who postulated that acute acute protection provided by opioids appears to be tightly linked to adenine receptors. However, the exact mechanism has been ambiguous. Works by Schultz and team in 2001 had observed the role of endogenous opioids in mediating ischaemic preconditioning and myocardial hibernation in swine laboratory models. The team summarized that that opioid receptor is mediating the effect of endogenous opioid peptides to elicit ischaemic preconditioning in rat laboratory models, rabbit laboratory models, and also in swine species. Its works by Chen and co-workers in the mid-90s who showed that naloxone modulated naloxone as pharmacotherapy modulated the actions of endogenous opioid peptides in a myocardial infarction mouse laboratory model. Chen and Van Winkle in 1996 had highlighted that naloxone had blocked PC or preconditioning or postconditioning in anesthetized rabbit laboratory models. In regard to this, it had been appreciated in as much that the opioid receptor antagonist naloxone had blocked several sort of receptors aligned to endogenous opioid peptides, and the human heart does actually have capacity to synthesize all three types. Indeed, at the same time, the cardiovascular system, or the human cardiac muscle or cardiovascular system, and endogenous opioids function has been noted to be complex by the fact that receptor expression is modulated by changes in across and around physiological conditions. In works that have had aligned in context of clinical practices that have been documented aligned to this. The National Service Framework, or NSF, for in around cardiovascular healthcare or cardiovascular conditions, interventions in recipients and patients with, for example, acute myocardial infarction, for example, should usually include, unless contraindicated, a form of pain relief, and to relate morphine has been documented as an opioid receptor agonist. Predominantly, it has a high affinity for the mu opioid receptor, like beta-endorphin, and it is also apparent that morphine appears to mimic endorphins during pain. In this regard, to works by Takasi and team in '99 have found that both morphine and post-conditional preconditioning protected adult laboratory had protected adult rabbit laboratory cardiomyocytes, and that these effects were blocked by pharmacotherapy and naloxone. The intrinsic opioid receptor system is complex, and receptors contribute to functional changes in normal and diseased myocardium. Beta-endorphin strongly relates to morphine, strongly relates to morphine, and with this and in this regard to opioid nature, and also has greater significance in human investigation, since it has a longer half-life in circulation comparing to enkephalins. Leu-enkephalin specifically is an endogenous agonist for receptors that are stimulated by alkaloids, and has multiple effects on the central nervous system and hypothalamus. Metenkephalin has been known metenkephalin is known to be involved as a pain perception, regulation, and memory of emotional condition and regulation around the mechanistics in around the immune system. Enkephalins also inhibit painful sensations by reacting with particular opioid receptors on sensory nerve endings. Has been documented to have in around 25% of the activity in the mouse vas deferens, and 30 to 60 times more potent than morphine in these species in murine or mouse models. Many laboratories have investigated serum levels of beta-endorphin under varying physiological pathological conditions, and beta-endorphin is exclusive and has a high affinity for the mu1 opioid receptor, and few investigations have assessed its role in human ischemic tolerance. It is also accurate to affirm that the majority of the studies are in laboratory animal models, and data confirming presence and activity of the mu opioid receptor in around human cardiomyocytes or cardiovascular in around human cardiovascular system or pathophysiology is and has been ambiguous. Animal models and localization studies indicate that mu opioid receptors are not present or expressed in cardiac tissue or parenchyma, and there are still no detailed investigation that assess beta-endorphin in pre or post condition or remote ischemic preconditioning in human investigations, although it has been generally identified that to an extent that beta-endorphin levels relate to pain in patients, for example where patients have clinical conditions such as angina. It's works by Falcone and team in 1993, where the team measured beta-endorphin in recipients of patients with ischemia and angina undergoing percutaneous transluminal coronary angioplasty. And this team, concluded or highlighted and concluded that beta-endorphin is higher and more stable in recipients of patients with silent ischemia during angioplasty, suggesting that endogenous opioid function during ischemia is associated with individual attitude toward pain. Beta-endorphin may have a role in cardioprotection through pre or post conditioning to protect myocardium during times of stress, for example by limiting the effects of particular inflammatory cytokines. Metenkephalins are products of the proenkephalin gene, and enkephalins appear to derive predominantly from the adrenal medulla, and are broadly distributed in the central nervous system. Similarly, similar to beta-endorphin, where this is similar to beta-endorphin, but methamphetamines act through both mu and sort of delta opioid mu and kappa mu and delta opioid receptors, and whilst works by Paradis and team in 1992, for example, who postulated there are increasing concentrations of enkephalins in ventricular rat laboratory models and particular parts of the laboratory rat tissue. Crucially, works by Halliday in sort of more retrospectively in the 1980s, 1983. Affirmed that in humans, adrenal enkephalins have a far shorter half-life than beta-endorphin, which may limit adequate concentrations from reaching brain centers to affect cardiovascular responses. Leu-enkephalin, also a product of of the proenkephalin gene, has a high affinity for delta opioid receptors than mu receptors. Dynorphin has an affinity for kappa receptors, and is broadly distributed in the central nervous system, including the spinal cord and hypothalamus. It works by Crumins in 1985, had demonstrated that delta and kappa receptors in adult laboratory rat heart models have been identified to an extent, but no evidence for mu receptors, and finally works by Dallagnone and team in 93 had reported high levels of beta-endorphin had been measured where silent myocardial ischemia had been identified or where it had been evident. So that closes in on this particular audio podcast in around beta-endorphin. Around this audio podcast. And in around the role of endogenous opioids in ischaemic preconditioning YPC in humans. This has been an audio podcast aligned to the Storm Robotics Soliloquy and a Storm Robotics production. This has been an audio podcast aligned to the series, the LabZtorm Effect v.3. Thank you.